So it is my great pleasure to introduce the first speaker for today, Professor Martyniuk. He's an associate professor at the Center for Environmental and Human Toxicology in the Department of Physiological Sciences at the University of Florida, Gainesville. He has more than 250 publications and is the internationally recognized expert in molecular neurotoxicology, focusing on defining adverse outcome pathways for mitochondrial dysfunction in zebrafish. Professor Martyniuk is the editor-in-chief in Comparative Biochemistry and Physiology, Part D, Genomics and Proteomics, and a counselor for the North American Society for Comparative Endocrinology. It is really my great pleasure to see you today, and I just don't want to delay your talk any further. Now it's your turn.
Great. Well, thank you so much, Magda. It's a very nice introduction. And thank you to RAMS and the group for the invitation to come here today and to tell you about the research that we do in the lab using zebrafish as a vertebrate model to understand pesticide-induced neurotoxicity. So I'm a toxicologist at heart, and I've become very interested over the years in how environmental pollutants affect animals and can potentially lead to neurodegenerative diseases such as Parkinson's disease. Okay, so this is just a brief outline of the talk today. I'm going to discuss some of the pesticides that are linked to Parkinson's disease. There's good evidence to suggest that they are, and how we go about studying those pesticides using zebrafish as an alternative vertebrate model for neurodegenerative diseases. We use this model to investigate mechanism, and I'll mention some of those mechanisms a bit more in detail as we go along. And a new kind of direction to the lab too that I want to talk a little bit about, because there is a lot of buzz, is the role of the microbiome in neurodegenerative diseases. And this link between pesticide exposure, changes in the microbiome, which could potentially be associated with Parkinson's disease. So we're just getting into this area of research, but I think it's a very exciting and very novel area to be pursuing right now in terms of environmental toxicology and neurodegenerative diseases. So this slide here really depicts the progression and the outlook of industry on a global scale. And kind of the larger the icons on this map, the more pronounced these industries are going to be over the next decade or two. And you can see that in almost all countries, especially in some of the developing countries, there is an increase in manufacturing, in pharmaceutical companies. There's an increase in oil and gas production. And the take-home point is that this is a global issue, that pollution is ubiquitous, and there's really no one part of the global scale that is responsible. It's really all of these industries are contributing to it, and there is no really kind of slowdown in terms of the chemicals that we will produce. So over the next several decades, like I mentioned, there's predicted to be an increase in the number of chemicals that are manufactured and used. And we use chemicals, we need chemicals. Chemicals are here to benefit and to improve our lives, but we have to be diligent and careful when we use these chemicals. And this is where environmental toxicology comes in, and the intersect between pollution and human health. So take-home message, chemicals are ubiquitous in the environment and are increasing really on a global scale. This was an article that came out in the Lancet a few years back now, but it's really quite striking, and it really puts the global pollution issue at the forefront. pollution has been suggested to cause more deaths worldwide than war or smoking or other diseases such as AIDS, tuberculosis, malaria combined. So it's predicted that pollution is the #1 single cause of early mortality events in humans. And it costs the public on a global scale $4.6 trillion annually. So about 6.2% of the global economy might be lost due to pollution issues. And again, nowhere in the world are you really protected against pollution. So again, we need to be diligent when we use these chemicals, and we need to understand negative impacts that can occur from using chemicals indiscriminately or ubiquitously. So this brings me to Parkinson's disease. I told you that these chemicals can be associated with a wide array of human diseases, and Parkinson's disease is no different. This is a disease with a long latency time, afflicts 2% of people aged 60 years and older. It's expected to double in incidence by the year 2030, cost Medicare in this country $23 billion annually, and is a significant problem. 15%, some estimates maybe a bit higher at 20%, tout that the risk of having Parkinson's disease is explained by 15 to 20% a genetic basis. So this leaves a large proportion or percentage of the disease that can be explained or influenced by other factors. There's very well-known, genetic mutations, alpha-synuclein, LRRK2, other genes that are significantly linked to an increased incidence of Parkinson's disease. But environmental factors play a huge role in whether or not this disease manifests. And it could be upwards of 75, 80%. So take-home message, the environment plays a very key role. So knowing this and having an interest in environmental contaminants and pesticides, we began some years ago investigating these links between pesticide exposure and Parkinson's disease. And I was kind of trained at the time when microarrays were big on the scene, and now of course it's next-gen sequencing and RNA-seq. But really at this time there was a lot of mechanistic data being produced, identifying or revealing transcripts and proteins through proteomics that genes and proteins were linked to pesticide-induced neurodegeneration and Parkinson's disease. And so these biomarkers of exposure and effect become very important, and they offer potential new routes of therapeutic treatments once revealed. So we compiled a lot of the transcriptional data for pesticides that had epidemiological links to Parkinson's disease in terms of exposures. And these ones here, so rotenone, paraquat, of course, is a well-known herbicide that's associated with an increased risk to Parkinson's, manib and dieldrin as well. There's epidemiological evidence to support a causative link between these pesticides and the disease, or at least initiates the disease. And through this analysis, we really kind of zeroed in on mitochondria as one of the major targets of pesticide-induced Parkinson's disease. So a lot of these pesticides that were associated with the disease really affected mitochondrial proteins and mitochondrial signaling cascades. And that's not to say that the mitochondria is the only organelle that's affected, but it does seem to be a major player in the disease progression. And so to study these kinds of events, we turned to the Zebrafish model for high throughput toxicity testing. So there is a strong initiative to undertake high throughput assays to predict toxicological effects. And so the Environmental Protection Agency applies such high throughput testing to assess whether or not chemicals are safe or whether they should be further examined in other vertebrate models. The reason we like zebrafish is that they're basically at a low cost to house these animals, and they're readily accessible. So we can house tanks upon tanks of zebrafish. There's a number of well-established transgenic lines that we've used and others have used to investigate mechanisms of pesticide action. And there's just really kind of no limit to how many fish you can hold in these tanks. So we typically hold thousands of individuals at a time. Another significant advantage of this model is rapid development. And so over the course of 3, 4, 5 days, these fish move from the embryo stage all the way into free-swimming larvae. So very, very rapid development, and it gives us an opportunity to capture certain stages of development in terms of pesticide toxicity. And we can follow these animals into adulthood. So we've exposed animals early on in development and put them in clean environments. And we're assessing whether or not there's these delayed effects on pesticide exposure. You can rear high numbers of individuals, and a number of mechanistic studies can be conducted in this species. It's got a sequenced genome and a significant number of tools that are available for it. And it's really a widely established vertebrate disease model at this point. There are zebrafish models for cancer, for diabetes, for Parkinson's disease, for Alzheimer's. And so really it's being applied to address a very broad number of questions related to human disease. So it's a very nice model to use. Following up on work that others had done previously, we optimized a mitochondrial bioenergetics assay using these zebrafish. So the nice thing about this model is that it's not a cell model. This is a living, intact organism for which we can study pesticide-induced neurotoxicity. So it really kind of gives us an in vivo early development perspective on some of these chemical exposures. We've also done cells too to correlate to developing zebrafish embryos, but this instrument here, very nice instrument. For those of you that are not familiar with this instrument, this is the Xfe Flux Analyzer. Very simply, it measures oxygen consumption rates of cells and organisms. This is very important, of course, because oxidative phosphorylation and the production of ATP requires oxygen. So this instrument can very quickly inform you on whether or not mitochondria are impaired because of a pesticide or are functionally normally. And this could be kind of an early warning sign for pesticide-induced toxicity. And this could subsequently lead to neurodegeneration. So the failure of the mitochondria to really produce enough energy to maintain development and cell viability. So we can measure whole embryos in this instrument. We have an assay for that. And basically this instrument will measure a significant number of mitochondrial endpoints. Some of these endpoints include basal respiration of the fish, ATP production. We can get a sense of maximal respiration and spare capacity. So if the mitochondria is pushed to a certain limit, is it able to go further? And that's a very important measure. Some studies have shown that a reduced spare capacity has been linked to adverse effects in cells and organisms. So I kind of equate this to stressing out the cell or the fish, the zebrafish, with a chemical, and then you place this organism on a mitochondrial treadmill and you measure how well it's able to produce and utilize oxygen. And from these measurements, we can assess again whether or not the mitochondria is healthy, impaired, or functioning relatively well. So this is kind of the big idea that we have, knowing that the mitochondria are central to pesticide-induced mitochondrial dysfunction, we will attempt to link pesticide exposures with mitochondrial failure, dysfunction, which can subsequently lead to oxidative stress, changes in hatch rate, and deformities ensue because of reduced ATP capacity. And for those surviving larvae, we can measure behavior. So another advantage of this model is that we can measure up to 96 individuals at a time with our behavioral assays. We do locomotor behavioral assays and anxiety-related behavioral assays. As we may know, Parkinson's disease is really part of the etiology is dysfunction in motor control, and we can assess that type of behavior with our high throughput behavioral assays. So this is kind of the framework for predictive toxicology. We want to predict whether or not chemicals affect mitochondria, and will this lead to behavioral deficits later on in larval fish or in adulthood. So a case study that we did is one using paraquat. Paraquat, very familiar, I'm sure, is structurally very similar to MPTP, which when converted to MPP causes dopaminergic neuronal degeneration. And this herbicide, which we have used extensively on weeds as a society in the past, has been shown to be epidemiologically linked to an increased risk of Parkinson's disease. This is a restricted use herbicide now. Because of this fact, we don't use this herbicide as much as we used to, but you can still purchase this herbicide in some cases. So again, structurally related to MPTP and can induce mitochondrial dysfunction in the neurons and lead to dopaminergic neurodegeneration. So a good model to understand mechanisms related to Parkinson's disease. We saw that paraquat impaired mitochondrial bioenergetics in the zebrafish, so again, supporting the idea that this herbicide can affect mitochondrial function in vivo. And we saw a significant decrease in maximum respiration of these embryo after 100 micromolar exposure. The chorion is this little shell around the embryo, which protects the embryo from environmental chemical exposures as well as environmental stressors. But paraquat was able to reduce maximum respiration in these fish. So associated with that was an oxidative stress response. We measured several transcripts related to oxidative stress and apoptosis. Superoxide dismutase 2 and heat shock proteins 70 were increased in expression. Again, kind of what we try to do is link the mitochondrial dysfunction with changes at the molecular level, knowing that these molecules are typically biomarkers for neurodegenerative processes. With the high throughput larval zebrafish locomotor assay, we observed hyperactivity, so kind of a hyperactive state with paraquat exposure. Here we have a sequence of a light-dark exposure. So basically larval zebrafish will move more during dark periods. This is a phenomenon called dark photokinesis, and we can measure their response in the dark versus the light. So in the light, they don't tend to move, but you can see that when you're exposed to this herbicide at 1 and 10 micromolar, in the dark zone here, this is the third string of light sequence, we see that there's a significant increase in hyperactivity or distance moved within these little wells. So each one of these little wells here has an individual fish, and we can track each one of these individuals over time to get a mean group. And we repeat these assays over 3 or 4 independent experiments to really validate our data. So again, trying to link that mitochondrial dysfunction due to pesticide exposure with downstream apical endpoints of importance like locomotion. So take-home message, pesticides can change oxygen consumption rates. We've also done similar studies with Manib and Xerum. These have also been epidemiologically linked to Parkinson's disease. We've observed several genes related to oxidative stress that are affected by these pesticides. And again, this can be associated with the mitochondrial dysfunction in many cases. And really what we've seen and others have shown is that zebrafish are an extremely useful model for studying neuroactive compounds, especially because you can use these high throughput assays to really increase the power of your experimental design and really test and screen different doses, different concentrations, different time points, because in toxicology, we all know the dose and the time point really kind of dictates the adverse effect that one would observe. So it's really nice that you can cover all of that with this high throughput model. Okay, so I'm just going to switch gears here and just tell you a little bit of the work that we've done with the microbiome in zebrafish. And perhaps it's no surprise to you all that the microbiome has really been established as a major contributor to human health. There's a whole number of different diseases and conditions and states that are influenced by your gut microbiome. And changes in diversity, richness, and function of the gut microbiome can be observed following different chemical exposures as well as dietary exposures, but chemicals can also affect your microbiome. So there's been a big push to really kind of elucidate those links between chemical exposure, microbiome, and neurodegenerative diseases, as I'll show you. So for these experiments, we used a chemical that is an organochlorine pesticide. This is in the same class as pesticides like DDT, which I'm sure you're all familiar with. Dieldrin is, all of these pesticides are banned because of what we know, but they are legacy pesticides. If I was to measure or assess your blood right now, I'd probably find dieldrin and DDT in it. Dieldrin is ranked number 18 on the disease registry database. So this is a pesticide that is significantly linked to a number of human diseases. And it's still ranked number 18 as one of the most concerning. Arsenic is number 1 in terms of chemicals that can cause serious health effects. Dieldrin accumulates in tissues, and these compounds have multiple effects on they're endocrine disruptors. They can affect the thyroid, the hormone system, estrogens and androgens, and they can also affect your immune system. So they're pretty nasty chemicals, and this is why we don't use them anymore, but they're legacy pesticides. They're still found in the environment and still in tissues. So there's been some really exciting work done isolating alpha-synuclein and gut endocrine cells. And there's been discussions about how this protein in the gut is, linked to Parkinson's disease. And there's been around for a while now a number of hypotheses about the migration of alpha-synuclein moving from the gut into the brain to cause those aggregates. So Parkinson's disease has been touted as a disease that may likely start in your gut. And so the question becomes, how do chemicals that are epidemiologically linked to Parkinson's disease, how do they affect your gut microbiota? Maybe that is the mechanism by which these neuroactive compounds act. It's maybe through your gut and not directly at the level of the brain, although these chemicals like dieldrin do accumulate in lipid-rich tissues like your brain. So the gut is a very interesting target for these pesticides. And so we had the question, we hypothesized that dieldrin would reduce diversity and richness in the zebrafish gastrointestinal system. And we fed zebrafish dieldrin for 4 months at a very low environmentally relevant dose. This is a dose that most wildlife would receive in the natural environment, and perhaps a dose that we would potentially receive by eating fish and through dietary exposures. So what we found was that after 4 months, the zebrafish increased their dieldrin in their bodies. There was no impact on body weight in the low, medium, and high, in the low and the high treatments. The body weight after 4 months didn't change, did not affect their growth, but the animals did accumulate dieldrin in their tissue. So there was a significant exposure, and the animals were being potentially affected by this chemical exposure. We looked at the gut pathology, and we had some histopathologists really dig into the gut and the brain, and take-home message was that there was really no significant change in any of the tissues in terms of gross pathology. So neither the gut nor the brain were really impacted by this environmentally relevant exposure to dieldrin. They look quite normal. Interestingly, we did not find any direct effect on microbial diversity nor the abundance of certain microbes, and there was no difference in alpha biodiversity index. So dieldrin didn't really appear to shift the overall diversity. The microbiome is diverse, whether it was from fish treated with a high dose of the chemical or a low dose or the control group. But that being said, we did identify some microbial taxa at the phylum and genus level and other levels as well that were affected by dieldrin. And just to kind of wrap things up here, this is a radar chart showing different phyla and classes that were measured and assessed in the gut microbiota. Those phyla and classes in red are those that were significantly shifted based on the dietary exposure. Green is the control group, purple is the high dieldrin, and blue is the low dieldrin treatment. And take-home message is that we did see some changes. For example, for MICUTs, there was a significant increase in the presence of these critters in the high dieldrin group. And one thing that you can do with, and this was done with 16S sequencing, but one thing you can do is some predictive, use some predictive algorithms to identify potential processes that are affected by the shifting microbial communities. So what we did is a technique called predictive relative metabolic turnover, or PRMT. So this again is based on 16S sequencing. It is an in silico algorithm and technique, but it gives you clues as to what processes are affected by the changes in your microbiome. And probably most strikingly is we found that selenium tended to be a major process that was affected. So methyl selenic acid, selenite, hydrogen selenide, and sulfonyl cysteine were all metabolites and processes that were affected by the dieldrin exposure. And so why is this relevant? Well, there's been some new studies coming out and some older ones too that really suggest selenium and other trace elements are very critical for the etiology of Parkinson's disease. Selenium is very, very important in the oxidative stress response. And if we're talking about mitochondrial dysfunction due to these pesticides, perhaps there is a link there between the mitochondria, the ability to protect against oxidative stress due to dysfunctional mitochondria, and this link to Parkinson's disease. So some very interesting literature. This is a new review that has recently come out. And so we're seeing changes in selenium in the gut microbiome following exposure to this pesticide. So in conclusion, dieldrin can be incorporated into the zebrafish tissue. These again are environmentally relevant levels, which is very, very important. As toxicologists, we don't want to aim too high and have a kind of a non-environmentally relevant exposure. Selenium dysfunction was related to the microbiomes, and for MICUTs appears to be sensitive to organochlorine pesticides. What's the significance of this type of research? Well, it really shows and supports that pesticides are associated with adverse effects and disease. And we need to understand the mechanisms of how neuroactive pesticides affect mitochondria to generate safer chemicals. And so a lot of these high throughput assays that I've discussed today can be used to screen chemicals for adverse effects on the mitochondria, for hyperactivity. And we have a lot more work to do, of course, but there's some terrific assays that are available due to this very amendable zebrafish model. So with that, thank you very much for listening. And I just want to acknowledge Andre and Hua here, who did much of the work with the microbiome and my lab as well. And I look forward to further questions. Thank you.
Hi, thank you for the great talk. And I just want to remind the audience to post their questions in the Q&A on the bottom of which you can see on your Zoom screens. And I would like to start with my like the first question which I have. Can you speculate what is the contribution or what is the link of your finding with microbiome to the aggregation of alpha-synuclein? How is it happening? Do you have any ideas that you have?
Yes, no, we're kind of at the early stages of exposing the fish to the different pesticides that have been epidemiologically linked to Parkinson's. And so knowing that there are certain microbiota that are sensitive to those pesticides, I think the next step would be to maybe dig down into that a little bit more. Of course, staining for alpha-synuclein in response to some of these treatments and how that protein responds to a change in gut microbiota, I think are kind of the next steps to tackle. There's been some nice work in mammals that have really kind of dug into that. But from the fish perspective, I think we're just at those initial early stages.
Yeah, that's that is a fair thing. My other question is how the microbiome of zebrafish compare to the human one of the mammals? Are there differences? Just to kind of orient us a little bit in the like bacteria which you're finding in their prevalence.
Yeah, so there are some pretty striking differences in certain phylum in terms of relative abundance. There are core phylum that are pretty consistent across the different taxa, but they, in terms of percent similarity, that's a tough question. But there are pretty, there are differences, pretty striking differences. So. Yeah, it kind of eludes me exactly how different they are.
Yeah, because I guess, I mean, it's one, but it still will give you an idea. And have you looked at if they are related of what you like the bacteria which are affected primarily? Probably you're currently identifying to which ones are corresponding in the humans.
Yeah, there's some similarities in terms of the mammalian literature, but yeah, we've always been kind of focused on the fish perspective. And so we do most of the comparisons kind of based on other zebrafish studies. And I mean, the mammalian literature does creep in, but yeah, there's definitely, and I'm trying to think, that paper was a year or two old, but we did identify some phylum and genus that were conserved across the taxa in terms of responding. But then I think, just trying to remember here, there was maybe one other paper that had done dieldrin exposures and looked at microbiota in rats. So again, when you're using different chemicals, the data is just not really there to make those types of comparisons. I've seen papers with a lot of the polycyclic aromatic hydrocarbons in microbiota. But yeah, to find those kind of to link up the pesticides that are epidemiologically linked to Parkinson's and have mammal studies and fish mammals, the literature is pretty sporadic. You know, people have done different chemical exposures looking at microbial changes, but I think, you know, that's a great thing, is to start to begin to piece all that together. But right now, I think it's pretty sporadic.
Yeah. That's really exciting. And I see a question in Q&A. This is from Christian Grissinger. The question is, for pesticides, it is not only toxicity, but also how the organism deals with them. Is there a possibility of to extrapolate from C. elegans to humans?
So yeah, so I mean, C. elegans is another widely used non-mammalian model for neurodegenerative diseases. It's not really used too heavily as a toxicological model, but it has some applications in that area of kind of pesticide-induced human disease. So, you know, I think zebrafish is kind of somewhere in the middle between C. elegans and humans in terms of, you know, that chain of making the jump to human disease. But I do think that these non-mammalian models can offer kind of some unique perspectives, and especially with C. elegans and zebrafish, really the power and the advantage is that you have that replication, you have that high throughput ability. And I mean, in terms of zebrafish and human, their genome may be 70–75% similar, depending on different reports, but somewhere in that realm. So you're talking about a genome that's 70–75%. So that might be close for some, that might be too far for others. But I think all of these models offer kind of unique perspectives.
Yeah. Thank you so much. So this is before we move to the next speaker. Oh, I just saw a question in the chat. I believe this is from Wang. What kinds of neuro-related behavior tests can you do in a zebrafish?
Yeah, so I think one of the most widely used is the locomotor activity tests. There are other tests for anxiety. So we've optimized an anxiety kind of anti-anxiolytic assay to assess whether or not the fish are anxious. They will respond differently to light and dark, and we'll want to explore those different kind of arenas in the plate, depending on how anxious they might feel after a pesticide exposure. There's a novel tank test for adults. So again, kind of more exploratory behavior to get at anxiety. When you put fish into a tank, if they're not anxious, they'll explore that tank a lot sooner than an individual who is anxious. We haven't done it, but other groups have done object recognition type tests, memory tests in zebrafish as well. So compared to, you know, the rat, there's a pretty good number of zebrafish tests that align with what's out there in mammals. Yeah, there's several behavioral tests.
All right. Thank you so much. And for the sake of time, we will be moving to our next speaker. Do you mind staying for the follow-up questions?
Oh, absolutely. Yeah, thank you.
Yeah, thank you. So, Matthias, yeah, you're doing it. Starting the slides, and while I'll just do the introduction. It is my great pleasure to introduce the second speaker for today's ZOOMinar talks, Professor Matthias Buck. And he is currently at the Department of Physiology and Biophysics, Case Western Reserve University. And he's also a director of Northeastern Ohio Highfield NMR Facility, Cleveland Center for Structural and Membrane Biology. So he did lots of moving for his PhD and postdoctoral studies. He's a native of Germany, but he did his PhD degree in the University of Oxford under the mentorship of Professor Chris Dobson. And after that, Professor Buck moved to the States for his postdoctoral studies, working first with Professor Karplus in Harvard and Professor Rosen in Sloan Kettering Cancer Center. The current focus of his research is protein-protein and protein membrane interactions in cell signaling and migration in organ development, cancer, Alzheimer's, and macular degeneration. His lab uses a wide range of structural biology, computational, as well as biophysics tools. And I will be not going to steal more of his time of his talk. Thank you so much for coming and nice meeting you.
Good afternoon. Happy Martin Luther King Day for those in the US. And thank you very much for Rams and his team for inviting me, and for Magda for the kind introduction. This is a ZOOMinar series I've been following from afar. It's been great, and I'm honored to be here despite 3 feet of snow in Cleveland over the last 12 hours. So now for something completely different, protein biophysics. I'll mainly talk about molecular dynamics, some NMR studies. The theme of the lab is biophysics of protein interactions as a molecular basis for mechanisms in cell signaling. 2 proteins interact, what happens and how. We're particularly interested in transmembrane receptors, which only cross the membrane once with the transmembrane helix. We'll come to that in a moment. The questions are, how does ligand binding outside to the receptor get transmitted through this helix to the intracellular regions, and how do these then interact with other proteins to transmit signals in the cell response. There's also ligand-independent signaling, which has become a major theme in some cancers with our receptors, where basically the cancer overexpresses the receptor so much that it leads to clustering and some form of activation, although often with quite different functions. As was mentioned, we pursue a bunch of structural biology approaches. I'll mention some NMR and biophysical measurements, and even some cell-based functional assays. Computational methods are key to the lab, main part of this talk. And I'll give a quick overview of 3 projects in the lab. The role of transmembrane segments in signaling mechanisms, domain dimer at the intracellular side of plexins and its complex formation with Rho GTPases, and then on the F receptor system, a small domain called SAM, how it forms heterodimers. And I don't have time to talk about our other theme in the lab, RAS, now interactions with RAF, although I might make a mention at the very end. So the receptors we are looking at are shown here: a plexin, a bunch of its co-receptors, receptor tyrosine kinases, famous in cancer, also neuropilin required for many plexin signaling mechanisms, and then the F receptor. When you color in the regions that have been solved by crystallography on the outside, and now increasingly with cryo-EM, and also on the inside of the cell. This is beginning to be reasonably complete picture, at least of some of the states, whereas around the membrane, it's still a bit of an unknown country. It's been very difficult to crystallize single transmembrane helices, even as dimers. NMR has been suitable for some of the systems, and simulations have, I think, really made a lot of progress in this arena. The basic idea as far as mechanisms, how these receptors come together and transmit signals from the outside to inside, concerns dimerization that brings these transmembrane helices together. But even within such dimers, there can be a sliding of the helices, change of their crossing angle, or their rotation that can occur and lead to different states. One idea that I think Bill DeGrado and I think perhaps others found a while ago is that transmembrane-like peptides can at times bind to these single receptors or dimers and change their signaling states. So, for example, we're collaborating with Dominique Bagnard at the University of Strasbourg. He found many years ago a neuropilin peptide which inactivates neuropilin and plexin signaling. So how does that work? We've done over many years a lot of all-atom simulations. They take quite a lot of computational resources, and more recently we've moved to coarse-grained simulations simply because we can get more done locally on our GPU cluster. So for all-atom simulations, it would take really a month or so to simulate microseconds, whereas coarse-grained simulations have a speed up of about 1,000-fold. That's because the representation is simplified. Instead of having all atoms in the simulation, we just have a few particles here with the Martini force field, and they have different properties, those particles. Recently, about a year, maybe 2 years ago now, Martini 3 was, 2 years ago as a beta also, but last year as the final version, Martini 3 was released. And we've done a study recently to help and validate that force field using these helix dimers, how they form in membranes. So we form idealized helices of the transmembrane segment, put them in, this is a DMPC bilayer, they're 60 angstroms apart and then can float around. They relatively quickly come together within a few hundred nanoseconds and then move around in these structures to try and find a minimum. We've compared the structures with those derived from solution NMR data, mainly from bicells. And those reference structures here are shown in yellow, whereas the structures we got, the major population is shown in different colors. You can see that we can get close to the NMR-derived structures, often within 1 or 2, 3 angstroms. Some are a little bit further away, and there might be reasons for that we're discussing in this paper here, which is currently under review at a journal. One thing we noticed, though, was that the structures are not really in a very deep minimum, but we can populate side structures and to convert with them in the simulations. So here is a population geometric measure is shown the interhelix angle at the center of, and the distance at the center of mass on the other axis, and you can see this spreads out into then one main configuration and some side populations for most systems. So that raises then the question: what determines these structural preferences and their interconversion? For example, here for the A1 and A2 systems, the A1 structure is preferred as a right-handed crossed dimer with 50 degrees, whereas the A2 structure is more in parallel. This is both experimentally and from the simulations. As an example of how we can look at specific interactions, Francesco Berrera at the University of Tennessee asked us to help his project with a 2 transmembrane-like peptide, which activates the FA2 receptor. He took the wild-type sequence and then changed the C-terminus, make it all glutamic acid, which gives the peptide a pH dependence to insert into the membrane when these become protonated. There are also some glutamic acids here in the membrane. Their pKs are shifted, so they're neutral, but they're polar, and this then creates different orientational preferences, how this peptide might bind to either a single native receptor helix or a dimer. And he asked us to test different versions here. Experimentally, when you just have the native receptor, the cells migrate in an assay through a gel, so-called scratch assay. Whereas if you stimulate the FA2 receptor with its ligand ephrin, now only 60% of the cells migrate. This is a repulsive response, the function of the F receptor. This can be mimicked by application of this the C2 peptide, basically without ligand, the receptor is being activated, whereas these other peptides don't work as well. In the simulations, we found they come together with the preformed dimer, or if you put all 3 peptides in the simulation, they form the same structure. In the case of the activating peptide, it docks to the side of a dimer here. So this is the peptide added in gray and white and black, whereas for the peptide that works less well, the M3 version, it tends to insert itself in the middle of the 2 receptor transmembrane helices, and this has been published recently in GMB. We can even see some details in the coarse-grained simulations. So I mentioned the pKs are shifted, so they're not charged in the membrane, of course, these glutamic acids, but they then do prefer close interactions with other polar groups, which there aren't really any in the wild-type sequences, but then they grab onto the main chain carbonyl or NH groups. So those interactions are seen here for the C2 peptide, the activating one, whereas the inhibiting one or non-activating one, it makes some other interactions here. So to put everything together, the experiments and the simulations, inactive states have either the transmembrane peptide added to a single helix or pushing 2 helices somewhat apart, whereas the activating configuration has the peptide added to the outside, stabilizing inactive form. I want to briefly talk about one aspect of the intracellular region of plexins. Moving back to plexins. It's a bit unusual as a protein because it has two segments that have high homology to GTPase activating proteins or gaps, but the region is inserted here, low GTPase binding domain, and we started the plexin project many years looking at this RBD. It gave very nice spectra, or so we thought, at high protein concentration until we started counting signals, and we saw that about a dozen signals were missing. Others were low intensity, so something was going on with the dynamics of the protein. And indeed, when we did gel filtration at these higher concentrations, we saw the protein was a dimer. At low concentration, it turns into a monomer, and indeed, using a cryoprobe, you can run protein at 10 micromolar or even below. And we saw these 12 missing signals. The other signals were very nice, similar intensity. So these signals here come from a monomer-dimer interface and just get broadened out because of monomer-dimer equilibrium and dynamics within the dimer. At the time, well, this is a reminder of time regimes in conformational dynamics in NMR with respect to the signals, the chemical shifts. So if you have an interconversion between two states on a slow time scale here, 10 per second, you observe by NMR both states, both environments of the proton, for example. When this becomes faster, there's a broadening of signals. Eventually they coalesce, but they're so broad they disappear into the spectral noise. As the interconversion becomes faster, your sampling is such that you see the average position of the NMR signal between these two states. So we just had an example of this intermediate exchange because the peaks were disappearing with the plexin RBD dimer. And as a strategy, this was published many years ago, we assigned as much as possible of the spectrum we had at high concentration. That left several areas here in white. And then we looked at those sequences. We argued that one might want regions with intermediate hydrophobicity because protein-protein interactions, you would likely want a mix of hydrophobic, hydrophilic residues. And then also, if those regions had biological value, they would be somewhat conserved. So we looked across the family, close family members across different species. Then we made mutations in these regions here and found that some of them stabilized the monomeric form of the protein. We're coming back to this project, even though it's been many years, because recently a new postdoc in the lab, actually a PhD from Ram's lab, discovered that there was interesting pH dependence in some members of the plexin family for their RBD regions. The crystal structure was then determined with the collaborators of the dimeric protein, and one of the long loops of the RBD forms this little beta strand where the residue we had mutated, tryptophan 1830, forms a critical interaction here with an arginine across the little beta sheet. And indeed, when we mutate this, we have a monomeric protein. So now we're puzzled right now about a pH dependence of a dimer to monomer transition going from pH 7 to pH 8, which we're trying to explain for some members of the plexin family. the RBD binds to Rho GTPases, and again, NMR is extremely useful here because we can carry out experiments with one protein being labeled, giving the signals. We're adding the GTPase RAC1, in this case unlabeled, and we see the effects this binding has on the resonances from the interface here in violet. They're in a different physicochemical environment, and so the signals will shift if the interaction is fast, relatively weak, or they will these peaks disappear and will appear at a new chemical shift in the case of slow exchange, which is what we have here. Even though that's true on the side of the RBD here, for these residues being in slow exchange, on the side of the RAC1, it's not so straightforward. There's a lot of regions in intermediate exchange, but some peaks here experience chemical shift changes in pink. Let's see, this is what I was, let me try that in a different way. Somehow the movies sometimes don't work directly in the presentation. I don't know whether people can see this. The number of crystal structures were solved with the complexes between the RBD here and GTPases. We did all atom molecular dynamic simulations, and you can see there's a lot of fluctuation, slight changes in the orientation of the two domains, relative loops coming on and off. So at any one time, really only 40% of the crystallographic residue contacts are maintained. We're talking about a dynamic protein complex, and this shows up in interesting ways. For example, if you do a network analysis, you look at all the fluctuations and how they're correlated. We see that in the case of RAC1 binding to this RBD domain here, we have almost no communication across the interface. They still behave as individual units dynamically, whereas in the case of round one binding, there's extensive communication, coupling of motions across the interface. I want to tell a very brief story about dynamics at the domain level that explained a property of plexin, the catalytic domain we discovered. So Rho GTPases bind here to the RBD that hangs off to the side, but the rest of this orange part of the protein is a GTPase activating protein, downregulates RAS and RAP1B. When we do a titration with these substrates, say RAP1B here, and we look at the amount of phosphate that is released, because as a gap protein, it hydrolyzes the GTP to GDP plus phosphate, we see a nice sigmoidal curve here with the wild-type protein. When we cut off this juxtamembrane region here or make critical mutations in it, we lose that cooperativity. And how can one explain this? At the same time, postdoc in the lab, Andrew Lee, discovered that one of the several crystal structures of the plexin gap region showed part of a loop here in red. He then modeled and extended it, which partially covered the active site of the protein. So what make the substrate, the RAP1B binding, more difficult. At the same time, in his simulations, this loop could swing back and bind to the juxtamembrane region here, explaining why that region is important for this cooperative behavior in the activity. And we were able to put all this together by looking at a paper from Raphael Brischweiler's lab here in PNAS a few years earlier. A nice comment by Vincent Hilser. They called this effect allocarry. It's a kinetic allosteric effect in an enzyme and doesn't require even multimers. You can explain it just with a monomeric protein here. The idea is that at low substrate concentration, the loop would block the active site, then swings back once in a while, substrate comes in, but then it leaves as a product, loop swings back, and you'd go over this process. Whereas in high substrate concentration, the moment a product leaves, the next substrate is able to bind, and the loop is still swung back. It doesn't have enough time to cover this because substrate comes back in early. So allocarry, story we published in, do I say it here? Yeah, Cell Molecular Life Sciences last year. The F receptor system is the receptor tyrosine kinase. What's unusual about this family of proteins is that below the kinase domain, there's little protein called a SAM domain. And these SAM domains are used to make heterodimers with other proteins, or even homodimers for some family members. In this case, for A2, it forms heterodimer with an enzyme SHP2, which breaks down PIP3 here and maintains the receptor longer in an active state at the membrane. So quite a few years ago, we determined the solution structure of this heterodimer. It's a 5-helix bundle. We used, again, chemical shift perturbations, orientational restraints from RDCs, as well as a handful of direct protein-protein contacts, NOEs, across the interface. And when you put all these restraints into essentially a docking calculation to satisfy them, we observed one main cluster of solutions, about 80% of structures in this cluster, and then 2 side clusters. Well, normally one would say, well, these are slightly high energy, don't satisfy the restraints as well. There are fewer, let's ignore those, but we had reasons to think that these might be interesting. What do these different configurational states look like? It's most easily visualized looking from the side here, and how this 5th helix in the SAM domain aligns with one another between the 2 domains. So you can either have them pretty well aligned or rotated out by 60 degrees or even further to 120. And underlying this are different patterns of salt bridges across the protein-protein interface. So we had some fun doing simulations here on Anton. This is still Anton 1 at the time, 2.4 microsecond of simulations to see whether these different configurational states would interconvert. And indeed they do. If you look here, they aligned on this A2 SAM domain, helix 5 here and helix 5 there. This one does transition between the NMR-like structures of at least 2 of these configurational states. The third one is only sampled very briefly. How do these transitions occur? We looked at diagrams like this, where we have all possible residue interactions on one axis, and then the distance between them on colored in here as a function of time. And we see that before you break one interaction, here you make a new one or an intermediary one in order to be able to walk around a diagram like this, convert between these states. And the closest analogy we can think of was this kind of monkey bar playground scenario where you definitely want to hold on with one hand, otherwise your proteins dissociate, and then to move around this ladder, you need several hands, several interactions. And again, then we looked at the protein dissociation process. We have mutated here some residues at the interface to make it less stable. And within a few tens of nanoseconds, the protein dissociates, not immediately, but after hundreds of nanoseconds of simulation first. And you saw this, well, maybe I might play it again. It's not occurring immediately from this tightly binding interface, but instead this other subdomain first rolls around in place, then goes to a different interface here before flying off into space. Even it dissociates briefly and comes back a little bit. When you follow this all with interface areas, you see the episode of it coming back briefly and in terms of energy calculation. So we asked quite in some detail why this comes apart, what are the contributions here. One is the solvation for energy, which becomes more favorable when the proteins dissociate, but also changes in protein dynamics play a role. I'll come back onto that later. We then wondered, why is it doing this with these destabilizing mutants, but not for the wild type? So the wild type, these microsecond long simulations, even if you include the more less populated clusters, it's really stuck in a minimum and can rotate around and so forth. But these mutants here change that energy landscape, so the protein is able to slide in one domain relative to the other one, here to a less strong interface. And it's from those interfaces that the dissociation of the protein occurs. It's known from the structures in the protein database that typically only 9% of the entire solvent accessible surface area here of a protein complex is involved in the primary interface interaction. That sort of shocked me initially when I saw that number. I thought it'd be a bit bigger. And then when you look at the character of this primary binding interface here in an open book view compared to the entire complex, you realize that really here there's quite a high concentration of charged and hydrogen bonding. I think there's one hydrophobic group here in the middle that doesn't show very well compared to the remainder of the interface, the remainder of the protein. So again, a dissociation occurs from more weakly binding interfaces, not directly from the tightly binding interface over here. We then wondered, well, what does this look like in reverse protein association? And the concept of energy landscapes, protein folding funnels applied to protein association dissociation, I think is very useful. So there's a kind of encounter complex that's formed and it's been characterized in some NMR labs. Then there might be alternate bound state conformations that, for example, this one is just the reverse of the previous movie, and so it had to first bound, but had to still rotate into the correct place at the last moment. When we ran all atom simulations, though, with proteins placed 45 angstroms apart. Now I know how this works. You have to press escape before you can stop the movie. Okay. We ran it with the same CHARM36 force field, even on Anton. This didn't help much because the domains came together very quickly, and then they were mostly stuck on certain parts of the surface and not in the wild type crystal structure NMR configurations. At the time, Robert Best and Michael Feig pointed out that the CHOM36 force field was slightly overemphasizing protein-protein versus protein-solvent interactions, and they made a correction to arginine side chain parameter. We scaled van der Waals water interactions by 5%, and this led to simulations being slightly different. For example, shown here, the association process is somewhat slower. It allows the proteins more time to pre-orientate themselves. And then once it's on the surface, even though it's an encounter state, the proteins are more able to roll around on the surface and even sometimes dissociate and rebind. So from a 7% success in simulations of 550 nanoseconds, proteins coming together correctly, we went to 18% success in this paper here. I'll skip this. So to step back, what can we conclude, speculate about the nature of protein interfaces? One thing is that they are multi-use. I think there's a molecular economy of protein interactions that you want to use the same interface with different binding partners, or the same binding partner but in different orientations. The alternative to have additional interfaces, say over here, over there, not so great, because anything that's unoccupied might lead to association, sort of amyloid formation and so forth. The other question is, why do you want different configurational states? And one answer could be that in the cell, actually, not all of these different states are allowed. Some might, for example, here clash with the membrane in geometrically restrictive environments. And finally, by having some more broader energy landscapes, more modest binding affinities, we form these complexes transiently, and they can come apart as is needed in cell signaling mechanisms. You're not stuck in a particular structure forever. I want to come back to the dynamic aspect of this kind of processes of dissociation and association. We wondered about how does the dynamics of the protein itself get affected by either being in a complex or being here in free proteins. And we did entropy calculations based on either order parameter treatment of bonds or vibrational analysis. And the picture that comes out here for dissociation of the A2 SHP2 complex is that entropy increases in both proteins when they come apart, especially in the A2 protein. It's all over the protein. Here at the SHP2, it's more at the binding interface. But we know what the total entropy changes from isothermal titration calorimetry and from the calculations here, just based on the protein dynamics, it's about 30% of the total entropy change. That can be validated experimentally. We did extensive measurements here of relaxation, the dynamics of the main chain and the two SAM domains individually, and when they're together, there's some changes here, but also on the microsecond, millisecond time scale, actually the dynamics gets completely quenched. So there are no more motions we can see on that time scale in the complex. which brings me to the last point. We collaborated, helped Carla Matos group in looking at a dimer of KRAS. They crystallized a dimer in presence of effector protein bound, again called RBD. We did simulations with that crystal structure as well as with the individual proteins, and this dimer, in absence of the RBD, did the similar entropy analysis and came to the conclusion here that this entropy change in making these two RAS proteins dimerize was quite large and unfavorable, whereas when the RBD is attached, the entropy change here is much less. Even though there's actually no conformational change communicated through the proteins, it seems it's a dynamically entropy favorable process now, because the RBD attachment here to the KRAS has already quenched, kind of prepaid the entropy loss that would be unfavorable. And the concept of then dynamically driven protein association says that, yes, if your protein needs to lose entropy when it forms the complex, this can be stimulated by binding of a ligand or here another protein to another interface, which pre-quenches or gives you makes this more rigid already. So the entropy change here in delta S2 is less. And conversely, one could do the opposite, of course, bind a ligand and make this more dynamic, and then this step would be more difficult. So to summarize, I showed you work in progress on the transmembrane-like peptides, how they interact with the transmembrane helix dimers of receptors, the FA2 example, Plexin, Rupilina progress. Talked about the Plexin Rho GTPase binding domain, its dimerization, and the NMR spectra. We get the mutations that destabilize the dimer we are visiting. The RBD Rho GTPase complex is considerably dynamic from simulations, and experimentally, certainly by NMR, we've had some challenges, have still not finished the structure of the complex. Plexin, a gap function, mechanism of allocarry, a kinetic mechanism that generates cooperativity, and then on the FA2 SHIP2 SAM heterodimer system, introduce this as a dynamic protein complex, a dissociation association simulations, and then computational. I didn't talk too much about experimental analysis of dynamic allostery, which we've seen in other. Actually, the Plexin, the RAC1 system has a nice example that we published that already in 2008. Then the principle of dynamic-driven protein interactions. That's we just resubmitted to structure, might be coming out soon. I want to thank members of my lab. Andrew Lee is no longer shown here. He started his own lab in Tianjin University. He did all the simulations, but now Anita Shahu is doing those coarse-grained simulations. Pravesh here is doing the F receptor project experimentally, and Chinmay, together with Giacomo, is looking after Plexin. I want to thank collaborators, Luca on the Plexin project, and especially Francisco on the F receptor project, computational resources and funding, and you for your attention and interest. I think I might have gone a little over time. Sorry about that.
Yeah.
Lunchtime is almost over.
No, that's fine. Yeah. Thank you very much for the nice talk. And again, just to remind to the audience, please post your questions in the Q&A section of Zoom. And I see that Rams posted 3 questions. So
Okay.
Rams, do you want to ask your questions? So I'll go read them.
Oh, I could ask if you want me to.
Yeah, please.
Nice talk, Matthias. Very nice collection of works. In the beginning portion, you mentioned about the TM-TM interaction. I wonder, do they? So I see that there are hydrophilic residues in the TM domain. How stable are the monomers to start with? And do lipids play any role at all in this oligomerization? And why do these monomeric peptides oligomerize to form homo-oligomers?
So the first question, what's known about the lipid property, the bilayer properties? Yes, they have a big role to play. So thickness, membranes, membrane raft formation. Francesco has a nice paper on PIP2 interacts a bit with the juxtamembrane region on the inside of the cell of the FA2 receptor. But because the PIP2 is associated also with membrane rafts, it has a different thickness, and it's more the membrane thickness than the JM region, it seems, that creates a different structure, because you have a different crossing angle preferred of the helices in the membrane. And there are lots of other very nice studies using cholesterol from John Straub and Chuck Sanders, and yeah, many examples. We are doing model systems at this point. We're not doing accurate eukaryotic membranes that have many components, and both simulation and experimentally, I think that might be the next step to get to even more detail. The second question, how are the TM monomers? Good question. I don't know what they would do. I presume that they'd be helical, because that's what satisfies most of the polar interactions on the backbone. But while the fluorescence measurements have shown monomer-dimer equilibria, so in terms of diffusion kinetics and FRET, so they must be stable and reasonably happy. Yeah, do they oligomerize into larger clusters? I think you can probably get up to 4 relatively easily with these receptors. Beyond that, I would think that the extracellular domains and intracellular domains, I mean, they're relatively close to the membrane in some cases, have some geometric blocking effects to allow the TM regions to come together. But of course, you could cluster them by the extracellular, intracellular domains and form a network quite easily, which is what believed to occur in the active state of the A2 receptor, the tyrosine kinase active state.
Thank you.
So there is a question from John Straub, and he says, very nice talk. In the case of EPHA2, I found your results on the dimer and trimer structures quite interesting. They are both hepta repeat and glycine zipper motifs implicated in EPHA2 dimerization. Can you discuss how these or other interactions serve to stabilize the dimer and trimer structures? And that question was posted on the chat if you want to.
Yeah, I'm also looking at it. Yeah, thanks, John. Very insightful questions. They're often coexisting gly-XX-gly and leucine zipper type motifs, and I think that's just a general theme, that these transmembrane signaling processes need to switch between different configurations, either within the dimer or changing, going from monomer to dimer to trimer. The question about trimers is intriguing because in the case of Plexin, we published a simulation study in 2015 with all atom modeling, and we modeled a trimer of the Plexin TM region and found that it seems to utilize a polar residue, a pair in the membrane. There's a threonine serine that makes nice hydrogen bonding between the helices. And so when Francesco's peptide had this glutamic acid, that's a polar thing too, and it again, it leads to trimers basically. So perhaps the trimers, well, are trimers compatible with GXXG motifs? I don't know, but certainly with Leucine zipper type things they are, because you can have the GCN4 trimers. Well, they are of course soluble, but the inner part, you stick a bunch of isoleucines together, I think.
Yeah, thank you. The next question is in Q&A from Carmelo La Rosa. Did you check if monomers in the dimer show symmetry with respect to the secondary structure?
Symmetry. Yeah, it hasn't really occurred in the Samsung case. In the case of the kinase domains, which that also had simulations and solutions, and the membranes we are thinking about symmetry. Also experimentally, it would be much easier if we would only observe one set of NMR signals. I think it's been discussed in the case of RAS dimers, for instance, that there's some symmetry, some asymmetry, how they stick together. And this goes on to Alex Gorfie's question here. He's asking about the RAS RBD interaction, and he's curious about the entropy results. Well, I can send you a preprint. I think it didn't quite make it to archive. It was part of a huge other paper that we broke up into smaller papers. But we should hear back from structure. I think, well, I can send it to you and you can have a look. I know that several people are interested in this topic. Sorry, I'm blanking now on the name. She also was going to put something together based on experimental data. No, I'm sorry, blanking on the name.
Yeah, thank you so much. At that moment, I think it will be like anybody who has questions to the two speakers today, please raise your hand so we can move you to the panelists. Ram, Sam, and Bikash, do you have any questions? One curious thing which I noticed is that Bikash Sahu, and I think you have, I was not paying attention if she's a graduate student, a postdoc. She also has last name Sahu, so I don't know whether they're related.
Maybe I'll ask her, but we have the phenomenon recently that we're able to attract good postdocs because I have enough colleagues around me in structural biology and in other interesting areas that often the postdoc is already married and they come with their spouse who's also a scientist. And now we have actually three couples in the same department. one of them is in my lab, and then in the surrounding lab, they're the spouses. So.
Yeah, that's actually a nice observation, because in science it's so difficult. So you need to kind of have the same to be able to have all these long hours.
Yeah, I only tried having a couple in my lab once, though, and that didn't work out too well.
Oh yeah, two in the same lab, it would be difficult.
That was many years ago, and I think it might have been connected with the personalities involved in my interaction with them. So
yeah, that is true. Rams, any questions which you have? And let's see.
No, I don't have
how
any
the
questions.
audience is doing. I have a question. I'm kind of ignorant about the receptors, but I saw that, and you kind of slightly, if you touch base about the VEGF receptor, right? And this is the one which is with the implication in ALS. Is that correct?
in ALS,
Yeah, because mutants, but sometimes when you have these abbreviations, I don't know if we're talking about the same. The abbreviations are exactly the same. Yeah, I saw at the beginning of your slide here.
That's news to me, but I should look it up.
I may be entirely mixing this up.
Plexin and neuropilin, they are implicated in Alzheimer's disease, and we put R21 on looking at one aspect of this, but
the one which I'm referring is vascular endothelium growth factor.
VGFR2. It's a co-receptor of neuropilin.
It's been implicated in ALS.
ALS.
Yeah, I don't know. Simply because we are on all this aggregation portion and stuff like that.
Well, it competes with plexin to bind to neuropilin. So I wouldn't be surprised if it eventually shows up also in Alzheimer's and Parkinson, because it'll take away from, and well, there's one report of plexin neuropilin complexes binding A beta from a Korean lab. And we were able to convert that observation and some modeling into a supplement to our grant. But apparently lots of things stick to A beta, so we'll have to see the details.
Yeah, that's
what it does to plexin signaling, for example.
Yeah, there is the very you're correct in that. I think we have another question from John Straub. I found your analysis of the role of charged residues in protein-protein interactions interesting. Have you considered the role of surface curvature as well as the charge in facilitating protein desulfation and protein-protein interaction? That's in the Q&A.
Oh, okay.
I don't know if.
So he's referring to curvature of protein-protein interfaces.
I just promoted John to the panelists, but we can go ahead and.
Yeah, I haven't thought about it. Maybe you can elaborate. Bears in mind. Hi, John. You're muted.
Hi, thanks again for a nice talk. Yeah, I was just I was partially thinking of this because Rams and I were just in a symposium honoring Xian Ham's work, and she had passed away a year ago, and
that was so tragic.
Really quite tragic. And I know you're aware of her work on looking at propensity for protein aggregation, and she had used a integral equation theory to look at solvation. and had some evidence that sometimes charged residues can play unexpected roles and hydrophobic surfaces and encouraging aggregation. And so I'm just thinking that sometimes, when we're thinking about the desolvation costs and how charged residues behave, sometimes it can be a bit complicated, and was wondering what other approaches in terms of evaluating the role of those charged residues on the surface. It was just brought to mind.
Well, one thing I again surprised me is that, and she made that point too through her work very elegantly, that even very tight complexes like Barney's bar star, there's like 20 waters still at the interface, basically bridging charged and polar interactions. It's not that things are very well desolvated.
Right. Yeah, interesting. Okay, thank you.
Chris, I have a question for you. Very interesting talk. In your pesticide effect on the microbiota, do you see, maybe I'm sure there must be studies on the connection to antibiotic resistance, maybe cancer and so on and so forth, because bacteria has a bigger role than just for Parkinson alone. So could you comment on the other areas where this kind of connection is established, maybe at the molecular level?
Very, very good question, actually. So that's actually a big movement right now in aquatic toxicology, is to understand the role of antibiotic effects on the microbiome, antibiotics that are found in water, but also antibiotics that are ingested by humans as well. So there's been a few studies now with different antibiotics looking at zebrafish microbiome, and it does change the microbiome pretty pronouncedly. Again, you're getting different classes different classes and genera changing with those antibiotics. So I think it's very complex. There's no kind of unique pattern per se at this point, but it does change the microbiota. Yeah, even just low levels in the water too. So that's enough to perturb it after 4 to 6 weeks of exposure.
Interesting. Thank you.
Yeah, thanks.
Yeah, and I also have a question for you, Chris, following on that. It's just out of curiosity, the bacteria which you find to be responsive, is it gram-negative or gram-positive?
Ooh, I'd have to go back and look. Yeah, it's basically those functional analyses will group different genera according to kind of their functionality of what we think we know about what the bacteria do in the body. So those functional predictions are based on several different species of bacteria, probably some positive, some gram-negative, that are related to that process. So for in our example with dieldrin and selenium, there's several different species that can contribute to those processes of generating or biodegrading selenium, and they can be both positive and negative. So yeah, I wish it was that simple, but it gets a lot more complex exactly who these critters are that are related to the process. But it could be upwards of hundreds of different species that are related to that process.
Yeah, thank you.